Well, thank you very much. And I really am grateful for the opportunity to join this group and also to present on Zanubrutinib for patients with previously untreated CLL, really focusing in on insights from the SEQUOIA trial. So we're going to focus. BTK inhibitors have been clearly a mainstay of CLL therapies for the last decade plus, and have been instrumental in really leading the charge of these landmark changes in CLL treatment paradigm in CLL, both in the monotherapy setting with initially ibrutinib and now acalabrutinib and zanubrutinib used in treatment-naive CLL and recommended across risk groups by NCCN guidelines and based on approvals. And we also know that the BTK inhibitors can be combined with BCL2 inhibitors, that these have distinct and complementary mechanisms of action and can be combined both safely and result in significant efficacy in CLL, including the treatment-naive setting, resulting in approvals and use currently in the clinic. So we're going to focus today largely on the SEQUOIA trial. And so this was a complex study, and so we're just going to spend a moment to walk through the protocol here. So there were four arms on this trial. First were arms A and B. So remember, these were treatment-naive CLL patients who met criteria for therapy and across different risk groups, including patients with and without deletion 17p and/or TP53 mutation, but there were differences based on which arm you were assigned to. So arms A and B were the randomization arm. So this was the sort of the pivotal study that ultimately led to the approval for patients with treatment-naive CLL without deletion 17p. This was an open-label study wherein patients were randomized to zanubrutinib monotherapy at 160 twice daily until progression or intolerance, or to BR, again with a primary endpoint of progression-free survival. Arm C was a zanubrutinib monotherapy arm for patients with treatment-naive CLL with the deletion 17p, again with a 160 twice daily dose. In Arm D, which we're also going to highlight today, was a combination cohort of patients with treatment-naive CLL with or without a deletion 17p or TP53 mutation who received zanubrutinib and venetoclax combination therapy. And there was an option for UMRD-based treatment discontinuation in this cohort. So for background, zanubrutinib was initially approved for treatment-naive CLL based on a pre-specified interim analysis of arms A and B that was presented in May of 2021. In this initial analysis, zanubrutinib resulted in superior progression-free survival when compared with bendamustine rituximab. Importantly, also presented at the time was the Arm C initial data showing encouraging progression-free survival and response data among patients with treatment-naive CLL with a deletion 17p. We're going to focus today on long-term follow-up data from all arms, from arms A, B, C, and D with the combination zanubrutinib venetoclax cohort. These are data that were presented in the last year at international meetings, including the 2025 ASH meeting, and also for Arm D based on a recent publication. So baseline patient characteristics are shown in this slide for all arms. And I just point out that this is fairly representative of what one would expect for a treatment-naive CLL population. There are notable differences, of course, across these arms, as one would expect based on differences in the eligibility criteria, remembering that arms A and B were patients who were considered unsuitable for FCR chemotherapy. So this is a slightly older population. And of course, we do not see deletion 17p or TP53 mutations present. We see in Arm C and Arm D higher incidence of high-risk features, of course, given that Arm C was defined based on deletion 17p for inclusion. Arm D, of course, was enriched for deletion 17p or TP53 mutation, as these patients were stratified to one arm or the other. Progression-free survival data are shown here for arms A, B, and C. Among patients with treatment-naive CLL without a deletion 17p who were enrolled and randomized to arms A or B, the progression-free survival Zanubrutinib. Zanubrutinib resulted in a sustained progression-free survival benefit when compared with BR. The 72-month PFS was 74% with Zanubrutinib and 32% with BR in this population. Among patients with deletion 17p who were enrolled and assigned to arm C, the 72-month progression-free survival was 64%. Overall complete response rates and overall survival data are shown in this slide for arms A, B, and C. You can see if you look first at the overall and complete response data, that overall response rates were higher with Zanubrutinib compared with BR in the treatment-naive CLL without deletion 17p cohorts, 98% versus 89%, and were also quite high in the deletion 17p arm C cohort at 97%. Overall survival was robust across the arms at 84 and 80% with Zanubrutinib and BR, and at 83% with Zanubrutinib and deletion 17p patients. Again, this was at 72 months, so long-term overall survival, demonstrating very robust survival outcomes across cohorts. Now let's shift gears and look at the combination data. So this was arm D, so Zanubrutinib and venetoclax in treatment-naive CLL. This included patients with or without a deletion 17p and/or TP53 mutation. Overall, Zanubrutinib and venetoclax resulted in a median progression-free survival that was not reached, but a 24-month progression-free survival rate of 92%. Looking by deletion 17p TP53 mutation status, we do not see, although this was not a randomized comparison, we do not see apparent differences in the 24-month PFS between these arms, which appeared quite similar. And again, the median overall survival was not reached. The estimated 24-month overall survival is 96%. Overall response and complete response data are shown here for the combination cohort. The overall and complete response rates were 97% and 48% overall, and did not appear different based on whether patients had a deletion 17p and/or TP53 mutation. Now remember, arm D did include the option for discontinuation among patients who achieved UMRD4 response after the initial 2 years. The median time to initial peripheral blood UMRD4 was 19 months among patients with TP53 aberrant disease and 11 months among patients without TP53 aberrant disease, demonstrating differences in the kinetics to achieving UMRD4 based on whether patients had a deletion 17p or TP53 mutation. The UMRD4 rates overall in the peripheral blood were 59% and were similar based on whether a patient had P53 aberrancy or not. Importantly, relatively few patients on the study actually discontinued therapy based on UMRD4 achievement. And although we do see that among these patients who discontinued therapy, we see sustained remissions in a majority of these patients. These data really don't give us clear guidance about the role of treatment discontinuation based on UMRD4 in this population. Safety data for arms A, B, and C are shown here. Again, if you look at these cohorts, and we're just going to really focus in on the Zanubrutinib arms for arms A and C, these data are consistent with what we've seen across other Zanubrutinib trials with expected toxicities, but there were no new or unexpected toxicities in arms A, B, or C. And in arm D, this was the combination of Zanubrutinib and venetoclax. Again, we see rates of adverse events that were consistent with the individual agents, both overall and when we look at treatment emergent adverse events of special interest. In summary, long-term data from the SEQUOIA study clearly demonstrates continued efficacy and safety of Zanubrutinib as an effective first-line therapy for treatment-naive CLL, including in patients with high-risk features such as deletion 17p. In arms A and B, we see that Zanubrutinib results in sustained progression-free survival benefit over bendamustine rituximab. And in arm C, which was solely for patients with a deletion 17p, we see that zanubrutinib results in durable progression-free survival outcomes, which appeared similar to those without deletion 17p from the arm A from the randomized comparison. Looking at arm D as well, zanubrutinib and venetoclax results in a manageable safety profile and clearly encouraging efficacy among patients with treatment-naive CLL, including in patients with high-risk features, including TP53 deletion or mutation. And we see that a subset of these patients were able to discontinue therapy based on UMRD4 status. Thank you.
Thank you, Jake, for this presentation and a wonderful overview of this data. And I think there may be different positioning of this drug in different places and what is available to patients or not. Maybe, Uli, you can give a little bit a flavor of the European situation.
Yeah, hi everybody. Well, thanks for the nice presentation. I think since we are talking about first-line CLL, the European position would be, let's give these patients a chance to have a time restricted treatment, which I think would speak for a combination arm with venetoclax, clearly. This is also in the interest of our payers, to be very honest, because I mean this is how we get the treatment through. So I think it's pretty clear for the non-mutated TP53 arms. For arm D, I think those are extremely interesting results. If we could get patients with a TP53 mutated or deleted disease into longer remissions when they reach MRD negativity, that would be a strong argument for using this type of treatment first line, to my mind. So those are just a few thoughts. I hope I caught the European flavor a bit, looking at Martin and Stefano.
Any other comments? Astrid, anything in Latin America?
No, thank you. I agree absolutely with what has been said. This drug is available in our countries, and I think if we're going to do continuous treatment, maybe the question will be whether acalabrutinib or zanubrutinib. But I agree that we're all trying to go on to limited time therapy, and this combination, this doublet, is a good option, and also a good option for TP53 mutated patients.
Can I ask? Yes.
Yeah, please, Sonny.
Yeah. Thank you, Jake. That was really, really great. And just maybe two questions. One is, for arm D, did they continue to collect toxicity information along with the PFS? Because I think what ends up happening is that a lot of times people will collect toxicity information during the time of treatment, but then stop once it's over. And all you get then is the PFS data. And so I'm just curious about somewhat of the later toxicities within the time frame. And then the other question is, maybe it goes to like patient preference. Some people will prefer just simply taking monotherapy without having to have the added complications of the venetoclax ramp up. And so how do you think about that?
So for the first question, that is a common issue in many of these therapies or studies comparing time-limited versus continuous therapies. With arm D, actually, it was less of an issue because relatively few patients actually discontinued therapy at that time point. And so this was one study where actually the combination arm, we do get continued toxicity profiling in the Zanu venetoclax arm as well. I share your feelings about patient preference is ultimately one of the most important factors here for treatment decisions in CLL. And, I'm struck by some of the recent data showing a normal expected survival for patients receiving ibrutinib or ibrutinib venetoclax who have treatment-naive disease, which, and so although my preference is similar, I agree, in Boston, we're very European here. So I agree with the European perspective that time-limited therapy is really the preference for most patients, in my opinion. But, when we have a normal expected survival, who am I to say that that's the only option? And so I think that for some patients, that clearly continuous therapy with something simple like a single agent BTK inhibitor is a reasonable alternative.
Maybe I'm here the devil's advocate, but I would like to mention that, Sonali, in our outpatient clinic, I also have another experience, which is if you ask the patients whether you would like to start smoothly, the vast majority is for one pill a day, full stop. However, these are exactly the same patients who, after 6 months, are saying, well, I would like now to stop treatment. And when you come back and you say, well, this is what we discussed a while ago, and said, oh no, I had a totally different understanding. So what I'm saying is, this is one of the reasons why in Europe this time-limited regimen is so popular. On the other hand, even in the scope of the German Austrian study group, we where we always were putting our money on to BCL2 agents. I mean, for the p53 mutated, it's still going there as a continuous monotherapy. And to be honest, when I heard the first results about it, I think that was arm C, if I'm right. I said, well, looks better than what we would have expected with other BTKIs. But now with the longer follow-up, I think this is really satisfying and good news for our patients, that even with p53 alterations, you're able to achieve these very long remissions. And so finally, my question to the experts is, specifically to Jake, did you, in this study, was there a subgroup analysis analyzing the prognostic impact of different mutations or alterations?
I'm not aware of that analysis being done, but that's something that we can try to get for you after the meeting.
Thanks.
Jen?
Yeah, something I just wanted to bring up that this trial doesn't really address, and which has become another complicated factor when we're choosing therapy for patients with CLL, is whether to do triplet or a doublet therapy, and particularly for these high-risk patients. If you're TP53 mutated and want a time-limited option, we've had the kind of luxury of having a triplet clinical trial option for patients, which has been attractive. And I know it's in the guidelines now that you can choose that. I'm curious how the group here might kind of approach an arm D strategy versus a triplet regimen if they had the choice, or maybe it has to do with patient fitness.
Any comments on that? I think this is an important question. Maybe I see that Mike was very courageous to wake up very early to join us from Australia. And our colleague from this part of the world knows this drug for a very long time because they have been involved in early clinical trial with it.
Yeah, so we've been very involved in Australia in the development of Zanubrutinib and very familiar with it. And I think, one of the main appeals has been how very well tolerated it is when compared against other BTK inhibitors. But to go to Jen's question, at the moment, our access in this case is largely limited to single agent treatment in the front line. And so for time limited therapy, we're still looking at ibrutinib and Ven in that context, which is very appealing. And I think the jury's out on p53 mutated disease, and in that context, I don't think I really know whether non-limited BTK inhibitor treatment is optimal or time-limited combinations are optimal. I just don't think I know that I know where the data is there. But I appreciate that routinely people are using non-limited BTK inhibitor-based treatment in that particular population. Unfortunately, I don't have access to a triplet in this context, so I can't directly answer Jen's question. But at the moment, I would say most common practice in Australia is in p53 mutated disease to use non-time limited BTK inhibitor-based treatment, and in other patients to use time-limited treatment. But many of my colleagues will happily reach for a ven-based time-limited combo, irrespective of p53 mutation status, on the argument that the jury's not totally out on that question.
Astrid, I see your hand raised. I don't know if this is from before.
No, this is now. No, I agree that I think access to this triplet is not so easy around the world. The results have been very good, but I'm not sure it can be widely applied everywhere. And also consider that having so many new options now and with such good PFS and outcomes, I think adverse events are very important when choosing within these. So there was a higher number of infections when using anti-CD20, and maybe that is something that we consider in some of our patients, especially older patients. So I think that is something that we should also take into account when deciding treatment.
Can I ask two things to Jake, and maybe you touch upon that very briefly? And the first one is, while it's very well tolerated, we do know that a few patients stop using the drug after a few years for different reasons, which can be, I'm not sure if these data are really collected in the trial, and that's what Sony said, but do you have any insights about that? And the part two of the question is, there have been a lot of discussions regarding the different mutations occurring with the different BTK inhibitor after exposure when disease progress, and how do you see in this context Zanu?
Yeah, so first of all, in terms of the failure profiles over time, we clearly see less discontinuation due to intolerance with the second generation inhibitors compared with ibrutinib. The reasons for discontinuation over time, tend to be consistent with what we've seen historically. I can say in the clinic, though, it's oftentimes some of these nuisance side effects. Patients who come off due to myalgias or arthralgias, or these reasons that are not necessarily the things that as physicians we worry about, but are things that patients are very concerned about. I think that the key question to me is severity of the AE leading to discontinuation, and the status of the disease at the time of discontinuation, leading to the decisions about what to do next. With patients who are in a long-term remission, at the time of discontinuation, often able to tolerate, for example, a treatment holiday at that point. The mutation profiles are interesting, and I think that we're learning more and more about this over time. I think that there's some lead time bias because much of the early data comes from patients receiving ibrutinib. These are different drugs. They have different kinomes. They have different effects on CLL cells, and we see different profiles over time emerging. I think there's more unknown than known about at this point about the specific mutations and whether that will impact on subsequent treatment activity.
Can I just make a little comment? Is that was used to have the 80 milligram pills, which were easy to manage when the patient had myalgia or arthralgia or a few things like that. Unfortunately, at least in the US, we had now the 160, and then it's very difficult when patients have side effect to manage lowering the dose. And I think to me, it's a really pity because, again, you have patients that are in excellent response, sometimes MRD non-detectable by flow or without having, sophisticated assay in clinical practice. And it was rather easy to, I have had patients that clearly had a major change in their quality of life when decreasing from 80 milligram. But I think it's a pity that this is not available anymore, I would say.